Welcome to Dr. DEEPA Dahake's NEET Biology, and in today's session we discuss about the cell division. And mainly, as you know, there are two types of cell division. First, mitosis, and second is meiosis cell division. In today's session we mainly discuss about the mitosis division. So let's begin with the session and pay attention that what are the types of cell which show the mitosis division, how the haploid as well as diploid cells undergoes in mitosis division, because generally the students know very well about the phases and identical features, but when we come towards the haploid body and diploid body, so they generally have a doubts और confusions होते हैं आप लोगों को. Some of the students ने मुझे से request की है कि मैम टेक द क्लास इन हिंदी प्लस इंग्लिश, बट मेरे लिए क्वाइट थोड़ा डिफिकल्ट होता है कि आइदर आई कैन टीच थ्रू आउट इन हिंदी और आई कैन टीच ओनली थ्रू आउट इन इंग्लिश. सो आई प्लान फॉर द हिंदी मीडियम स्टूडेंट ऑल्सो. सो इन कमिंग वीक, maybe if it is possible with my time schedule, so I will try to upload the videos for Hindi medium students also. So those students also stay continue with channel for the Hindi topics and Hindi videos in Hindi mediums. ठीक है. So let's we begins with the mitosis division, और इसमें मैं try करके देखती हूँ कि whether I explain the things in both the languages or not. Mitosis division, as we study, mitosis division mainly you remember take place in vegetative cells. Firstly, you remember the mitosis division is take place in vegetative cells of the body, और vegetative cells को हम commonly कहते हैं somatic cells. So somatic cells of the body. So in vegetative cell or somatic cells, it is take place. First ये याद रखिए. Now whenever vegetative cells and somatic cells are undergoes in mitosis division, so because of that you remember growth and development body में complete होता है. So mitosis you predominantly remember mitosis division is responsible for growth and development of the body. So this part you remember very well. Body में जो growth and development हो रहा है, that growth and development, though the body is plant or animal, in both the bodies you remember growth and development mainly किस division के through होता है? Mitosis division. So responsible for growth and development of body. Now यहाँ पे देखिए, when we study about the mitosis division, so commonly we say mitosis is equational division. So commonly it is called as equational division. क्यों कह रहे हैं आप equational division? Because in resulting daughter cells the chromosome number remain constant. Constant means if the diploid parent cell undergoes in mitosis, so the resulting daughter cells are become diploid, and if haploid cells undergoes in mitosis division, then the resulting daughter cells remain haploid in terms of chromosome number. That's why it is called as equational division, as chromosome number become remain same भी आप कह सकते हैं. Chromosome number remain same in daughter nuclei or in daughter cells. This part you remember. Now the next point you remember when we study about the mitosis. So in mitosis you remember how many cells are produced upon mitosis. This also important. So you remember upon mitosis division, introductory part हम discuss कर रहे हैं. Upon mitosis division you remember two daughter cells are formed. This part you remember, and where we study the mitosis, if in microscope, under microscope or in laboratory. If you want to study the mitosis, so which material is best for the study of mitosis division? This question also asked. So here you remember root tips of onion. Root tips of onion are best material for experiments of mitosis, or if you study the mitosis under microscope, prophase, metaphase, anaphase, telophase, then commonly we use the root tips of onion. Now why we use root tips? So you recall the zonation of roots. In roots, when we study the zonation, so the meristem zone or zone of division is present in root tip, and that zone of division show the continuous mitosis division, because the mitosis, regular mitosis in plant is an identical feature of the meristem cells or meristem tissue. That's why we commonly use the root tips of the onion. So this you remember. Now when we come towards the mitosis division, so what exactly mitosis? This we all discuss about the introductory part discuss कर रहे हैं कि commonly इसको आप कह रहे हैं somatic cell division, equational cell division कह रहे हैं, and secondly division किसके लिए responsible है? For growth and development of body के लिए responsible होता है. When we study about the mitosis division, so you remember it is a type of division in which the chromosomes are equally distributed in both the nuclei or in both the daughter cells, because the chromosomes in one of the phase undergoes in duplication. So here you remember mitosis is a type of division in which duplication of chromosome is take place, and because of duplication of chromosome, you remember duplication of chromosome followed by equal distribution of chromosome in both daughter nuclei, or daughter nuclei will proceed to the development of daughter cells. So this much you remember. Now we come towards the phases of mitosis. When we come towards the phases of mitosis, so phases when we study, so predominantly mitosis division or every division you remember, it is completed in two phases, that is karyokinesis and cytokinesis. That means first the nuclear division is completed, and when nuclear division complete hoga, after that the division of cytoplasm is take place. That division of cytoplasm is called as cytokinesis. So here you remember in phases, first phase is karyokinesis, that is the division of nucleus, and second phase is called as cytokinesis. ये part याद रखिए. Karyokinesis, that means simply you remember karyokinesis means the division of nucleus. So nucleus division को आप कह रहे हैं karyokinesis. And cytokinesis, that means the cytoplasm is divided. How does the cytoplasm is divided? That we study when we discuss about the cytokinesis. Karyokinesis, you remember, predominantly karyokinesis is completed by four phases are there. Firstly, prophase. Second, metaphase. Third, anaphase. And fourth, you remember, prophase, metaphase, anaphase, and the fourth phase is called as telophase. So this part you remember. If any of the students have any kind of confusion for its sequence, so simply you remember PMAT. PMAT अगर आप याद रखते हैं, so you can recall the sequence of the phases, that is prophase, metaphase, anaphase and telophase. And once the telophase is completed, so upon completion of telophase, two nuclei are formed and then after the cell undergoes in cytokinesis. Cytokinesis is entirely different in plants and animal because एक major difference है in plant and animal cell, that is in plant cell wall is present, whereas in animal cell only cell membrane is present as a outermost boundary. So for that you remember when cytoplasm is divided. So in plant cytokinesis you remember it is take place किसके through complete होता है plant में, it is take place by cell plate method. तो plant में आप study कर रहे हैं cell plate method, whereas in animals we study the commonly cleavage method, or animals also show the cytokinesis by cell furrow method. So this part you remember very well, that the cytokinesis is completed in plant by cell plate method and in animals by cell furrow method. So this part you remember very well. Now यहाँ पे just first we discuss about how the diploid or haploid both the cells can undergoes in mitosis. Because when we study about meiosis, so in meiosis you remember, in meiosis only diploid cells are undergoes. But in mitosis you remember, in mitosis when we study, so here you remember in mitosis both the cells, haploid cells can undergoes in mitosis, as well as diploid cells or diploid parents are also undergoes in mitosis. When will be the haploid cell? First you see the example. First in example you remember algae, bryophyta and fungi. Clear? These three groups, algae and bryophyta, belong to planty kingdom. Lower plants we are commonly saying, as well as fungi. Separate kingdom is there according to 5 kingdom. So these members of these three groups show the haplontic life cycle, or they show the haploid body, and haploid body when they have. So their haploid cell आप याद रखिए, haploid cells of their body undergoes in mitosis during growth and development, as well as gametogenesis when take place. Gametes आप कहते हैं कि gametes are always haploid. So when the body is haploid, parent body, gametophytic body आप कह रहे हैं. So when parent body is haploid, so gamete you remember, gametes are simply formed by the mitosis division. But when parent body is diploid, as you appear in angiosperm plants, as well as in humans and in animals आप study करते हैं, diploid chromosome number is present in each and every cell. That means the body is diploid. And when this diploid body undergoes in gametogenesis या gamete formation के process में जाते हैं, so diploid organisms form the gamete by meiosis division. But haploid cells or haploid individual, that is algae, bryophyta and fungi, always form the gamete by the simple division, that is the mitosis division. So this you remember for haploid cells and the diploid cells. Diploid cells you remember, the diploid plant body present in pteridophyta, that is commonly called sporophyte. After pteridophyta आप study कर रहे हैं gymnosperm, angiosperms, as well as animalia kingdom you remember, or animals you remember. So angiosperm as well as in animalia or in animals simply you remember, including human too. Diploid cells are present in body. So during the growth and development, diploid cells are undergoes in mitosis. Gametogenesis, one of the most significant part that you will study in class 12th syllabus. In haploid individual, ये part आप याद रखिएगा. When in haploid individuals, gametogenesis is take place by mitosis. Because we discuss here mitosis, so we only talking about the gametogenesis and mitosis. जब हम meiosis discuss करेंगे, at that time we see how does the diploid individuals play significant role in gametogenesis by the meiosis division. ठीक है, तो this much you remember. Now we first study about the various phases of mitosis division. That is, what are the important feature. Here I am not going to discuss about the detail that early prophase. If we study about the prophase, so early prophase, late mid prophase, late prophase, that we are not going to study because we mainly study for the revision purpose here. So here you remember, यहाँ पर हम mainly identical properties are studied for the prophase, metaphase, anaphase and telophase. So let's begin with the identical properties of the prophase. So students, let's begin with the features of prophase. Clear? Pro means primary. So the first phase of mitosis division is called as prophase, and whatever we study about mitosis division in these four phases, mind में clearly याद रखिए कि we only study about the changes which take place inside the nucleus. Cytoplasmic changes हम यहाँ पे include नहीं कर रहे हैं because we mainly study about the karyokinesis or nuclear division. So first thing you clear in mind, prophase identical feature when we study, or normally when we study about the features of prophase. So in prophase you remember first we study the prophase begins with mainly condensation of chromatin fibers. So you all are knowing, and in nucleus chapter or the nucleus topic when we discuss in cell and cell, the unit of life, in that session also we discuss that the nucleus, resting nucleus or interphase nucleus show mainly the fine network of chromatin. Now slowly these chromatin fibers undergoes in condensation, that means chromatin become a chromonema condense to form chromatid, and then further condensation result in the appearance of the chromosome. So condensation of chromatin network or chromatin thread, you remember. Second feature you remember, slowly in prophase, in animals predominantly, this feature we only discuss किसके लिए कर रहे हैं आप, animals के लिए, because centrosome are only present in animal cell. So in animals you remember, or in animal cell, centrosome और pair of centriole आप कह सकते हैं. Centrosome start to opposite polar movement. So opposite polar movement you remember, opposite polar movement is start by the start by centrosome during the prophase. Early prophase में ये generally आपको मिलेंगे changes. Then in mid prophase, centrosome nearly reaches to the opposite pole and they will result in the formation of ester. So the ester formation is take place that we will study after the prophase phase. And then you remember, at important feature, this feature is generally asked for your competitive exam in true and false type of statement based question. At late prophase you remember, mainly identical feature, nuclear membrane and nucleolus start to disappear. This part you remember. So slowly nuclear membrane and nucleolus start to disappear and centrosome clearly reaches to opposite pole. And secondly you remember fourth phase, at late prophase also one another feature, that the chromosomes initially when the prophase begins, so chromatin network is appear, but at the end of prophase chromosomes start to appear clearly. This part you remember. Now what is happening initially when you appear? So the initially cell show the nuclear membrane, nucleolus as well as the fine network of the chromatin is present. But once the cell enter in prophase and the prophase nearly completed, at that time you remember inside the cell nuclear membrane start to disappear. So we only show the dotted appearance of nuclear membrane. Nucleolus also nearly disappear, so you forget about the nucleolus in late prophase. And the chromosomes, you remember, chromosomes are start to appear clearly. For example, here we taking that the cell which is undergoes in mitosis division, it shows the diploid chromosome number 4. So 4 chromosomes are clearly appear in thread-like structure. Different color or different patterns you use for easy detection. So this part you remember very well. So 4 chromosomes clearly we appear and nuclear membrane. Secondly, the centrosome, you remember centrosome in animal cell. Centrosome now reaches to opposite pole, and from these centrosome star rays like structures are appear. These are called as astral rays. So what is astral rays, that we will study. So this part you remember, and as well as the spindle fiber are start to appear at the late prophase. So this part you remember. These are the features of the prophase. Once as the nuclear membrane and nucleolus disappear, immediately the prophase will change into the second phase. That second phase is called as metaphase. So ये part याद रखिए. The first line of the NCERT for metaphase is one of the important line, because it indicate that how can you differentiate between the prophase and metaphase. So that line you read clearly. अब metaphase when we study, so in metaphase you remember identical feature again we study, formation of metaphasic plate. So metaphasic plate और metaphasic plate को ही commonly आप कह रहे हैं, commonly it is called equatorial plate. So equatorial plate or equator plate हम कह रहे हैं, equator plate is formed. Second feature, all chromosomes with their centromere arranged at equator plate. This is the identical feature. This feature also asks that identical feature of the metaphase under microscope. How can you identify that cell is undergoes in metaphase? So all the chromosomes with their centromere arranged at the equator plate or metaphasic plate. Secondly, important feature of the metaphase, the phase is used for counting of number and the study of morphology. If you count the number of cell in number of chromosome in cell, so for that you use only one phase, that is metaphase. So you remember this phase is used to study number of chromosome as well as morphology of chromosome. This you remember. Now we come towards the structure. Clear? So when we come towards the structure of the nucleus or cell for metaphase, the rounded shaped cells slowly now start to appear in oval shape. The diagram which we draw here, even in NCERT, the diagram which is provided for mitosis, that is predominantly for animal, because the centrosome is appear inside the cell. So here you remember centrosome now develop these star shaped rays are called as astral rays. This is centrosome. Both combinedly called ester, and then the spindle fibers are produces and these spindle fibers show this is the equator plate is formed, and then this much you remember. Every time the 3 spindle fibers are more as compared to the chromosome number. Now these spindle fibers are attached to centromere with the help of kinetochore protein. So here at that stage you remember chromosomes now clearly appear in the form of chromatids. So 2 clear chromatids you can visualize in each chromosome. So all the chromosomes you remember exactly present at equator plate. So this you remember 4 chromosomes clearly you appear. Okay, this plate you remember, this plate is exactly formed in center, so it is called equator plate, and 2 esters are formed in animal. That's why you remember in animal cell you remember amphiestral mitosis is there. Amphiestral means 2 esters are formed. That's why it is called amphiestral mitosis. In plant, centrosome is absent. That's why ester formation is completely absent. So because of that in plant, anestral mitosis we are saying. So anestral mitosis is present in plant. When you come towards the structure of one chromosome, so here you remember this is one chromatid, this is another chromatid. Here the cap-like structure is present, that is called kinetochore, and the spindle fiber whenever attached, they attached with this kinetochore protein. Spindle fiber never attached directly to centromere. They always attach to centromere with the help of kinetochore protein. So this part you remember. Now after that you remember metaphase is completed and then cell enter into the third phase. That third phase is called as anaphase. The identical property of anaphase in mitosis, because in mitosis we study the duplication of chromosomes take place. So duplication means actually the splitting of centromere or splitting of chromosomes take place, and because of that splitting both the chromatid of the chromosome become isolate or get separated. So anaphase you remember, characterized by the splitting of centromere, or the splitting of centromere is also called as the duplication of chromosome, because 2 chromatids now become separate from each other. So this is called as duplication of chromosome, and because of that the 4 chromosomes get converted into 8 chromatids, and now these 8 chromatids behave as a chromosome due to their content of DNA, and the 4 chromatids now start to move towards the opposite pole. So first you remember for anaphase, second character of the anaphase, that due to shortening of spindle fiber or contraction of spindle fiber, chromosomes, that is in the form of chromatid, chromosome start to move towards opposite pole. So this part you remember. So 4 chromatids as a chromosome, they start to move towards upward pole, and the 4 chromatids, 4 we are saying because as an example we take diploid chromosome number is 4. So here in anaphase you remember what is happening. The 4 chromatids, now the equatorial plate is nearly disappear, and the spindle fibers are become shorter due to the contraction, and the chromosomes start the opposite polar movement. This part you remember. So these poles are moved towards the opposite pole. Diagrams take small time. So this you remember again, and here also. In this way. Clearance of 4 chromatids as a chromatode. Why? Because initially the diploid content of DNA will be here. Diploid से कितना हो गया था 4C content. Due to splitting of chromosome, the every chromatid become 2C, and in terms of DNA, now the chromatid become equivalent to the chromosome, and because of that the chromatids, or you can say chromosome, start to move towards the opposite pole. So at the end of anaphase or at late anaphase, nearly the astral rays and the very short spindle fibers are there, and chromosomes, you remember, nearly reaches to the opposite pole. And here you simply I draw the 4 thread like structure for explanation purpose. So 4 chromatids, you remember, reaches to the opposite pole, and once they reaches nearly to opposite pole, that means spindle fiber now start to disintegrate. Then the anaphase will convert into the telophase. So now here we study about the phase telophase, and then we come towards the cytokinesis, that how the cytokinesis take place in plants and animals. So now we study about the features of 4th phase, that is telophase. This is the last phase of karyokinesis, you remember, or nuclear division. It is characterized by, you remember, nuclear reorganization. itself one term sufficient है telophase को explain करने के लिए, that nuclear reorganization, that means 2 nuclei are formed in one cell and one nucleus is developed at each pole. So nuclear reorganization, that means nuclear membrane and nucleolus start to reappear. First thing, because of that 2 nuclei are formed and one nucleus at each pole. So simply you remember whatever features we study in prophase, opposite to that features are take place in telophase, because in prophase, late prophase we study the nuclear membrane and nucleolus start to disappear, but here the nuclear membrane and nucleolus start to reappear. The chromosomes which are appear up to the anaphase, now the chromosome become again like a thread structure, or they comes in terms of the chromatin, you can say the chromosome become convert into the network of the chromatin. So this part you remember, and at the end of telophase you clearly appear here, you remember that 2 black chromosomes we are showing and 2 red chromosomes, fine thread like structure, nucleolus is formed as well as nuclear membrane is start to appear. So at the end of telophase 2 nuclei are formed, centrosome is present, but astral rays and spindle fibers are completely disappear. So this is the feature for the telophase. At the end of telophase, when the telophase is completed, immediately after that the process of cytokinesis take place in most of the cell. अब यहाँ पे देखिए, here it is decided कि whether the multinucleate cell is formed or whether the uninucleate cell is formed. कैसे decide होगा? यहाँ पे देखिए if the cell either यहाँ पे two condition है, either cell undergoes in cytokinesis, that is cytoplasm is divided, and because of that the cytoplasm, cell wall or cell membrane, whatever to be there in plant and animal cell, that we study in cytokinesis, but due to formation of due to division of cytoplasm, two cells are formed, and these two cells showing the presence of single nucleus, then the division is called as cellular division. And here you remember the cells are said to be uninucleate. But in case the cell cytokinesis is not followed, cytokinesis, you remember, cytokinesis not take place, and again nuclear division start. So what is happening? Inside a single cell, a multiple 4 nuclei, 4 से 8 होंगे, 8 to 16, a large number of nuclei are present because the nuclear division is not followed by the cytokinesis, and because of that such kind of multinucleate cells are formed. Multinucleate cell are commonly called as syncytial in case of animals. When we study about the locomotion and the structure of muscle fiber, at that time we study the muscle fiber or sarcoplasm of the muscle fiber is multinucleate or syncytial. So this is, you remember, this is the multinucleate cell. तो multinucleate cell कैसे form हो रहा है याद रखिए. Multinucleate cells are formed when the nuclear division or karyokinesis not followed by cell wall formation or cytokinesis. But when it is formed by cytokinesis, then the monokaryotic or uninucleate cells are formed. So now अब हम study करते हैं, come towards the cytokinesis. So here you remember, cytokinesis का meaning clear है आप सभी को. Cytokinesis का meaning है simply cytokinesis means the division of nucleus. ये part याद रखिए. Secondly, when we study about the division of nucleus, तो यहाँ पे division of nucleus I am saying the division of cytoplasm. Again है ना, slip of tongue. So cytokinesis, you remember, cytokinesis is commonly called as division of cytoplasm. In plant and animal, we clear cut have idea about plant cell and animal cell. So the process of cytokinesis extremely differ or totally differ in plants and animals. How does it take place in plant and animals? That we study in plant, as we already discussed in the form of chart. In plant, cell cytoplasm divided by cell plate method. So this part you remember. And in animals you remember, it is take place by cell furrow method. Major difference is there because in animals cell wall is completely absent. So the students who are having the basic idea about the difference between plant and animal, they can easily understand this difference. Now when we study about the cell plate method in plant, so cell plate, you remember, how does the cell plate formation is take place? Cell plate formation आप याद रखिए किसके थ्रू होता है? Cell plate is formed by phlegmoplast. The students who are new for the session, they can go through the cell, the unit of life sessions videos में, this video in which we discuss about the structure of Golgi body. When we discuss about the structure of Golgi body, so and function of Golgi body, so over there we study that one of the function of Golgi body is the formation of phlegmoplast. So by phlegmoplast, cell plate or initial state of the cell wall, cell plate is formed. Now what is cell plate or what are the phlegmoplast? Here you remember, this is the cell of the plant. Simple two nucleus, very rough diagram of nucleus I am making here. एंड आफ्टर दैट फ्रेगमोप्लास्ट पे कंसंट्रेट करिएगा. द वेसिकल्स ऑफ गोल्जी बॉडी, vesicles of Golgi body in small groups arranged exactly in the center of the cytoplasm. These vesicles or the group of vesicles is called as phlegmoplast. So for objective purpose you remember phlegmoplast is formed by, so it is formed by the vesicles of Golgi body. What does it contain? So these vesicles you remember, these vesicles contain the calcium and magnesium pectate, because as these vesicles are burst, so the calcium and magnesium pectate become hardened and it will result in the formation of cell plate. In future, cell plate become a middle lamella in plants. So middle lamella, we always say middle lamella is made up of calcium and magnesium pectate. So initially phlegmoplasts are formed, and after that when the vesicles which contain calcium and magnesium pectate, these vesicles get burst. After that you remember the cell plate formation is take place, and later the cellulose and hemicellulose get deposited. ये exactly center में नहीं आया है, so I rough this. So cell plate is formed here in this way, and the site where the cell plate formation is not appear in future, these sites become function as a plasmodesmata in plants or cytoplasmic connections we are saying commonly. So this is the common method in plant for the division of cytoplasm. When we come towards the animal, so in animals you remember only, simply you remember the cell when completely show the nuclear division. After that the plasma membrane from center slowly develop a furrow, and this furrow gradually extend कहाँ पे हो रहा है? From periphery it extend towards the center. So slowly this furrow extend towards the center, and after some time both the cell get separated from each other and the nucleus become occupy the center position in animal cells. Reason behind that, the vacuoles are more in number but smaller in size. In plant, nucleus is present towards the periphery because a presence of single large vacuole is the identical feature of the plant cell. So this you remember, that very easy part is cytokinesis. So this you remember for the cytokinesis phenomena. With this we complete the mitosis division. Importance अगर आपको आता है, so introduction में हम discuss कर चुके हैं importance का part. So in importance you remember mitosis mainly normally in diploid individual as well as in haploid too. Mitosis responsible for the growth and development of the body, predominantly vegetative cells में ही होगा. But in haploid parents, the body where the parent body when the haploid. So in haploid body, along with growth and development, this mitosis play a significant role in gamete formation also. So this part you remember for the mitosis division. In case अगर आप लोगों को कोई doubts हैं इसके बाद भी, तो आप लोग फ्रीली मुझसे पूछ सकते हैं. टेलीग्राम की लिंक्स नीचे प्रोवाइडेड है डिस्क्रिप्शन में, सो यू कैन ज्वाइन माय टेलीग्राम आल्सो फॉर द नोट्स एंड मोर अपडेट्स. So over there also I will provide the notes and objective questions for the exams. So you can take help on Telegram also. Okay. The students who are new for the session, they can go through the previous videos. If you like those videos and my sessions are helpful to you, in that case you subscribe my channel and like my videos. Like करना नहीं भूलना है, जो भूल जाते हैं आप लोग नॉर्मली. सो लाइक करना नहीं भूलना है, वीडियोस को लाइक भी करना है. एंड आफ्टर दैट अप टू नेक्स्ट सेशन में अगेन वी डिस्कस वन ऑफ द मोस्ट इम्पॉर्टेंट, दैट इज मियोसिस डिवीजन. वहां भी सेशन थोड़ा लंबा हो सकता है आपका, बिकॉज दीज टॉपिक्स वी कांट स्किप इन मिड. वन टाइम में वन टॉपिक वी हैव टू कंप्लीट इट. So in next session we discuss about the meiosis division. and up to that you stay motivated, work hard and stay blessed.